morning, dear students. We are starting another chapter, that is cell division. What is cell division? Cell division is the division of a parent cell and the generation of new cells. Through cell division, that organism can attain growth. It is cells reproduction. In eukaryotes, cell reproduction are of two types. They are mitosis and meiosis. Through mitosis, the formed daughter cells are identical to the parent cell. But in the case of meiosis, the formed daughter cells, they are not similar to that of the parent cell. In what aspect it is different? With regard to the genetic content. If we can take the genetic content of the daughter cell, it is half that of the parent cell. Next is cell cycle. What is cell cycle? Cell cycle is a cyclic orderly sequence of events that results in the formation of daughter cells from the parent cell. Okay, that is cell cycle. Orderly cyclical events that lead to the generation or division of that parent cell. And that cell cycle primarily have two major phases. They are interphase and mitotic phase. Mitotic phase, otherwise called M phase. Then we are going to see each phases. First is interphase. What is interphase? Interphase is a period of non-dividing cell. Even if it is not dividing during that stage, then also it consists of so many stages. Interphase itself consists of so many stages or phases. That we are going to see. Three phases are there in interphase. Which are the G1 phase is there, S phase is there, and G2 phase. So next we need to understand what is happening during each phase. Okay, first G1 phase. G for gap. So G1 phase, it denotes first gap phase, first preparatory phase. What all process is happening during the G1 phase? During the G1 phase, all the synthesis of required carbohydrates for the cells and required lipids are forming during that period, and that cells genetic content need to undergo replication. For that, so many proteins and RNAs are required, and all its synthesis are happening during this G1. So these are the main process that is occurring during G1, first gap phase. What about its duration? Regarding the duration, we can tell a correct time period. It is variable. That duration is variable with regard to different types of cell. And at the end of this G1 phase, some signals will come and trigger that cell to pass to the next phase, that is S phase. Then we are going to see S phase. S for synthesis, synthesizing phase. What is synthesizing in S phase? During the S phase, that DNA replication is taking place. And we already have learned that DNA or that chromatin have different levels of organization. For that, histones are very necessary, and that histones are synthesizing, and its histone mRNAs are synthesizing during this S phase. So DNA replication is taking place, and before that itself, the cell need to finish the production of histones and histone mRNA. Histone mRNA, what it means? Because histones are proteins. Proteins are formed out of RNAs. Which type of RNA? Histone mRNA. That's what histone mRNA is production also required there prior to the DNA replication. Then duplication of centrosome happens, Centrosome are two points that we can see at the two poles of the cell, and on around that centrosome mitotic spindle are organizing during the cell division. That all you will learn later. Next is G2 phase. G2 means I already told you G denotes gap. Second gap phase, G2 phase. And it is characterized by increase in the nuclear volume. Why that nuclear volume increase? Because DNA already replicated. So that itself can add some more volume to the nucleus. So nuclear volume has increased during the G2 phase. Then what all process happen during this G2 phase? They are a doubling of cytoplasmic organelles. All the organelles that present in the cytoplasm, mitochondria, like which all organelles, Golgi apparatus, mitochondria, centrioles, all those things need to be doubled. Only then they can equally distribute about the newly formed daughter cells. Then to ensure whether the DNA replication is complete, that also need to be ensured. And for further DNA condensation, because after finishing the DNA replication, again we already have learned that chromatin need to condense. For that so many proteins are required, and those proteins, whether it is synthesized or not, that also need to confirm. And also happening in G2 phase. So this picture shows the different stages in the cell cycle. We understand now, we already understand about the G1, S and G2. Next we are going to see the M phase or mitotic phase. During this phase, the nuclear and cell division is happening. What is the duration of this cell division? Length of this cell division, it varies from cell to cell. In the case of those cells that its requirement is more for bodily functions, those cells, its duration of cell division will be very short. And in the case of some other types of cells that its requirement is very less, then they have a lengthier cell division. So in that way, the duration vary. For example, for fast dividing cells like stem cells, stem cells of blood cells, then our skin cells, all those are having very short cell cycle because its requirement is more. And in our body, there are certain cells that are not at all. They are dividing. Example, nerve cells, muscle cells. Because we already have read and came to know that whenever a damage happened to our brain, it cannot be rectified because that a mature nerve cell, it will not undergo division. If any damage happened to that brain, it is permanent. The only solution is that the nearby or the neighboring nerves need to take over the function carried out by those damaged nerves. That only the solution. So, example for those cells that will not divide anymore, that include mature nerve cells and muscle tissue. Those cells will not divide when it attain maturity. There exists some other group of cells. They initiate their cell division only upon receiving some trigger of induction. Examples are liver cells. We have heard that if a person suffer from some liver damage, the only solution for him to have a liver transplantation from others. And in the body of that donor person, he suffer a lossage for a part of liver, and that lossage acts as a trigger to initiate the liver. It's his liver cells for next round of cell division. Okay, I think it's clear to you. Next is G0 phase. G0 means nothing. Nothing is there. When this phase occurs, it is an intervening resting phase. Resting phase where in G1 phase. During the G1 phase, sometimes an inactive resting phase occurs. that is G0 phase. Why it occurs? Because as the cell doesn't require a proper signal to proceed to next phase, that is the S phase, then that cell enters this G0 phase. Then we need to understand about which cells are having shortest cell cycle. One example is the early embryonic cells of Xenopus laevis. I will write Xenopus laevis. Sorry, yeah. In Xenopus laevis, during its embryonic time, its embryonic cells need to multiply very fast. As the cells need to multiply very fast for to facilitate growth, its cell cycle need to be very short. And for making that cell cycle short, what is the only optional way is to avoid the gap phases, or to shorten the time of the gap phases. Which are the gap phases? We know that gap phases are G1 and G2. So in the case of this organism, the time duration of G1 and G2 are very short, and the M and S phase alternate very fast, very quickly, to facilitate the faster cell division in the case of embryonic cells of Xenopus laevis. Okay. Next, we are going to see about the cell cycle control system. We know that for every process in our body, if a regulation is not there, then there is chance to have some mistake. Similar to that, cell cycle also possesses a control system. And that control system is called checkpoints. On reaching that point, the cells will halt for some time to check itself, to see whether any mistake happened during the process that it just passed. That is checking during that point. And if mistake happened, then the cell managed to rectify that before pursuing to the next stage. Okay, clear? Again, I will give a definition one more. Checkpoints are certain points at which the cell cycle halt until conditions means if any mistakes happen, that cell need to rectify that is the condition, until conditions are favorable to enter to the next stage. Okay, then which are the conditions that leads to have that checkpoints? First one is the chromosomal DNA damage. Suppose think that a cell is going to enter the S phase. We know that during S phase, that DNA will replicate. If a damaged DNA undergoes replication, the formed or the replicated DNA also carry the same damage. So, before undergoing that replication, the mistake in the DNA need to be rectified. So, it is very necessary to have a checkpoint to clear that damage. So, that is the first condition that lead to have that checkpoint. Next one, improper DNA synthesis in S phase. Next checkpoint can have after the S phase. After the S phase, we know that the DNA replication will maybe finished, and it is getting ready to enter the next phase, that is the G2 phase. Okay, and before entering that phase, the cell need to ascertain that the DNA is very correctly replicated. So, if it is not correctly replicated, then also that cell have another checkpoint. Then the third condition is the misalignment of chromosome during M phase. And after finishing that S phase and G2 phase, the cell is entering the next phase, that is M phase. M phase, we know that during that phase, the chromosomes are chromosomes are equally dividing and distributing to the daughter cells. That is happening in the M phase. And for correctly distributing, the spindle fibers need to correctly bind with the chromosome. If that alignment doesn't happen correctly, that led to another checkpoint to correct that mistake. Okay, I think that conditions are very clear to you. Then we need to understand. We are going to learn about the checkpoint exists for controlling the cell cycles. Which are the checkpoint that present in cell to control the cell cycle? Three checkpoints are there. First checkpoint is G1S checkpoint. Second one is G2m checkpoint. And the last one is Spindle checkpoint. First is G1S checkpoint. Listen to that term G1S checkpoint. G1 we know that it is the first gap phase before the S phase. So from the title itself it is clear that that checkpoint is between G1 and S phase. And once a cell passes this checkpoint, if a cell passes this checkpoint, it can enter which phase? S phase. As the cell passes this G1S checkpoint, it can enter the S phase. Okay, and in all checkpoints, two things are very necessary. Two factors are very necessary. First one is a factor to give signal. To give signal, for that a factor should be there, and second one a regulatory factor. To regulate this factor. Two factors are present in all checkpoints. Which are they? First factor give signal, and second one is a regulatory factor that regulates this factor. Both are proteins. So that point you need to keep in your mind while moving from one checkpoint to another. Okay. Then which are the factors need to check before pursuing to the S phase? First thing, need to confirm whether the cell attain the required size to undergo division. Whether the cell acquire or attain the required size to undergo division. That need to check before pursuing to S phase. That is the first step. Next thing, need to confirm that the required energy reserves are there to undergo division. Required energy reserves are there or no, that need to confirm. The third one, whether the positive signal to pursue to the S phase is there or not, that also need to confirm. What is that positive signal? I will explain to you next. Then the other thing that the cell need to confirm is that the DNA is intact or not. Is it damaged or not? That also need to check before entering the S phase. Okay. Next we are going to understand which is the protein that is giving signal here in this checkpoint. I already explained to you that in each checkpoint a protein is there to give signal, and another protein is to regulate that signal giving protein. Next point we are going to see which is that signaling protein present in G1S checkpoint. Its name is cyclin dependent kinases. Cyclin dependent protein kinases. This is the protein that is giving signal to the cell in this checkpoint to proceed to S phase. And which is the regulatory protein here? That regulatory protein here is G1S cyclin, because it is regulating that protein that is giving signal to the cell to proceed to the S phase. So its regulatory protein's name is G1S cyclin. It is mentioned here. What's the relationship between this cyclin and this cyclin dependent protein kinase? The relationship is that as such this protein is inactive. Alone it cannot do anything. It need a companion. That companion is this cyclin, G1S cyclin. Okay, I am just going to draw that protein that is giving the signal, that is CDK. Alone it is inactive. As it get a companionship of this G1S cyclin, as this G1S cyclin come and bind with this CDK, then it become activated. It become activated and it give signal to the cell to proceed from that G1 to S phase. And as the cell proceed from this G1 to S phase, then this signal is no more required. So because of that, then the level of this cyclin will reduce. Its level is reduced, and as its concentration dwindle, it separates. It alienate from this CDK, and the CDK loses the ability to give further signal. Clear? Okay, I hope that it is very clear to you how that checkpoint is happening, what all things are checking during that checkpoint, and after checking how it is getting the signal from CDK kinases, and what is the peculiarity of the CDK. The peculiarity is that it alone cannot do anything. After companionship with this cyclin, it become active and it give signal for the cell to proceed to next phase. After giving that signal, again the level of G1 cyclin reduce and it departed or detached from the CDK kinases, CDK protein kinases, and it lose its activity. Okay, that is G1S checkpoint. Next is G2m checkpoint. G2m checkpoint means it is giving the consent for the cell that cell is ready. It is fit to proceed to the M phase. So during that checkpoint, what all things the cell need to check that it is efficient to move to the M phase. The first thing that cell need to check is that every process that need to be finished during G1 and S phase is finished or not. That I need to check. Second, I need to check about the intactness of the DNA, whether the DNA is perfect, is there any mistake. That also need to check. Third point, it need to check whether the DNA replication is complete or not. That also need to check. Then next we need to know about which is the protein that is giving signal here, and also which is the regulatory protein here. That also we need to understand. This protein that is giving signal here is MPF protein. MPF protein. MPF protein means M phase promoting factor. This is also a CDK, cyclin dependent protein kinase. And this is the protein that is giving a signal for the cell to pursue to the M phase. Then which is the regulatory protein here? Regulatory protein is also a cyclin. Its name is G2 cyclin. It is otherwise called mitotic cyclin. Mitotic cyclin. It give activity to this MPF, this cyclin, by binding with this MPF. After this G2 cyclin come and bind with this MPF, it become activate and give cell the signal to pursue to the M phase. After the cell moves to the M phase, its level decreases and the activity of MPF stops. One more point I forgot to tell you during this G1s checkpoint, that is why the cycling got that name, because during the cell cycle, according to the need, its level increases and decreases in a cyclical manner. That's why it got this name. Cyclical manner means after the requirements as the cell proceed from one stage to another, after receiving that signal, then there is no need of this cycling. That time its amount decreases in a cyclical way. So that's why it got that name, cycling. Okay. Next checkpoint is spindle checkpoints. As the name implies, it's check whether the spindles are correctly attached to the sister chromatids. That is checking here mainly. It is a junction between metaphase and anaphase. It is the checkpoint between metaphase and anaphase. During metaphase, the chromosomes are correctly arranging in the equatorial plane of the cell, and during anaphase, the chromosomes, sister chromatids, are dragging to opposite poles. That is happening. So this checkpoint is happening between these two phases. Which all things are checking during this checkpoint? It ensures whether all the chromosomes are correctly aligned in the metaphase plates itself. And also it check whether the spindles are correctly attached with the sister chromatids. And also it check whether the spindles are intact or not. These are the three things that is checked during the spindle checkpoint. And here also one protein is to give signal. The protein's name is APC, that is anaphase promoting complex. It is a multi-protein complex. It gives signal to proceed to anaphase. Then after a cell proceed from M phase, means metaphase to anaphase, then there is no need of this APC. And this APC directs the degradation. After a cell proceed from metaphase to anaphase, this APC directs the proteolytic degradation of the mitotic spindle. Only then the next stage, telophase, can happen. Telophase and cytokinesis can happen. Okay, that is spindle checkpoint. Then what is G0 phase? G0 phase, I already explained to you, it's a non-dividing intact phase, and there is no growth stage, and some cells permanently rest in this phase, G0 phase. Example for such cells are muscle cells and nerve cells. They permanently stay in this G0 phase after attain maturity. Okay, with this we can finish today's class and section. Thank you.
I'm an icon, man. And my girl about a 10. I should fuck her in a Benz.